Good morning. Thank you to SelectBio for running such a well-organized conference as usual, and for the invitation to speak today. Methods for characterizing extracellular vesicles are evolving, and new technologies are being developed all the time that deliver size, concentration, and now fluorescent phenotyping with different degrees of success and different advantages and disadvantages. In this presentation, I'll review how some of these new and cutting-edge technologies work, compare their strengths and weaknesses, and share how we at Spectradyne see the technology landscape evolving next. In this talk, I would like to encourage you to think about three things. First, that EV size and concentration measurements are important, and they need to be taken seriously. Second, I'd like you to ask yourself, is my measurement method doing a good job? Is it working within the bounds of its natural limitations? And three, what kind of technologies do we need to see next? What are the needs for your applications? So I'll start with a review of why EV size and concentration are important, then of current methods for quantifying EVs, including Spectradyne's technology. And then we'll look at emerging methods, three new types of EV characterization technologies, and summarize with a discussion of what Spectradyne thinks we need to do to move forward. First of all, EV size is important, and this is important for a few different reasons, and in particular when we're looking at EVs as potential therapeutics. In this case, size determines the diffusion rates of the particles and also the clearance rates from the biological system. That is therefore determining the bioavailability and ultimately the therapeutic efficacy of the therapeutic, of the product itself. Secondly, size also determines the particle volume. And if you think about this, this means that it's controlling, it dictates the payload volume that can be encapsulated in the particle. And this is essentially the single particle dose. So how much active ingredient is being delivered per particle. Size is therefore critical for safety and efficacy of any nanoparticle-based therapeutic, and particular EV-based therapeutics. The concentration itself is also important because it's the overall dose, right? So for example, if we're delivering a therapeutic with EVs, then the concentration of the particles tells you the amount of material that you're administering. That's the dose. That's obviously important for safety and efficacy, as well as well-controlled science. Concentration is also important in sort of a more basic science experiment context as well. So for example, suppose we're studying EV regulation in response to a stimulus in two different systems. In this case, the concentration is a critical experimental variable. So a researcher might apply a stimulus to two different animal models, for example, and want to compare the EVs that are produced in each system. And in order to do this in a quantitative way, it's absolutely critical to control for the concentration of the EVs that are being measured, right, before we quantify with proteomics, for example. So doing so carefully enables a researcher to reduce their experimental variability and ultimately achieve clear outcomes faster. So essentially, good science requires precise normalization by concentration. One other example I'd like to highlight is when we're using EVs as a biomarker. So in this case, as an assay readout. In this case, the concentration of the particles really is the test result. And so the better we can measure the concentration, the higher sensitivity and specificity of the assay we can develop. And this ultimately leads to faster and more effective assay development. So accurate concentration and size are both critically important. Why or how are people measuring them currently? Well, the most common gold standard method is cryo-EM, right? This is the gold standard for imaging. Of course, we all know it's not practical to have one in your lab all the time. Another one is NTA. This is an optical technique. I'm sure you're familiar with it, and it has really helped develop the EV field since it was initially put out there, invented. I think nowadays researchers are coming to accept coming to understand its significant limitations at this point. We're looking for something new, which is part of this talk, right? In terms of non-optical techniques, there's TRPS, tunable resistive pulse sensing. At Spectradyne, we're a fan of electrical methods, so we like that one. But the specific implementation of TRPS is lacking a little bit, makes it hard to use. That leaves us with Spectradyne's technology, microfluidic resistive pulse sensing, or MRPS. This is a powerful technology that's fast, affordable, and easy to use. And let me tell you a little bit about how it works. Shown here on the right is our NCS1 instrument. This is a new technology, and it delivers accurate concentration, absolute concentration, and size, starting at about 50 nanometers up to as big as 10 microns diameter. And it uses electrical detection. It's a non-optical technique, and that makes it a very important addition to the toolkit for characterizing nanoparticle samples, including UVs. It's fast and easy to use. Typically, results are obtained in a few minutes of analysis. And it's easy to adopt. It uses microfluidics, and in particular, one advantage that we get from using microfluidics is that only 3 microliters of the sample are required for analysis. Let's talk about how it works. So MRPS uses essentially the Coulter principle. It's a modern implementation of the Coulter principle, which has been a gold standard for measuring large particles in cells, for decades. If you've had your blood sampled for total blood cell count, you've already benefited from this technology. So in this method, particles are analyzed one by one very quickly, and we measure the concentration and size of the particles in the most direct way possible. Let me explain. So we flow all the samples through constriction in the fluid flow, and every time one particle goes through this constriction, we get an electrical signal. And from this signal, we measure 2 main things. One is how big the particle is. That's determined by the size of the peak here of the event. And the second one is how quickly it passes through the constriction. And that tells us the flow rate of sample through the constriction, and ultimately provides the concentration information, because we know the volume of sample that we've been counting particles in. So this is a single particle measurement. If we measure for longer, the data looks like this. And this example very quickly shows, many particles being detected of 4 different sizes of particles, right? There are 4 bands of amplitude here. We can see that directly from the count histograms of particle amplitudes. But we can do better than that, because we know the sample volume that was measured. We can convert these counts to concentration, absolute concentration. So when we do that, this is the kind of plot that we get. This is for the analysis of the data I just showed you. And this is a typical output for our technology. It's got concentration on the y-axis and particle diameter on the x-axis down here. This particular sample was a mixture of polystyrene beads of 50, 90, 120, and 150 nanometers diameter. And you can see that the NCS1 is able to resolve each of the components of the sample and be quantitative over any size range here that you wish. So we can measure the concentration of each of these components or of what's in between. So this combination of accurate concentration and size, together with the ease of use of the method, are just not available on the market currently anywhere else. So let's turn to the second question now that I want you to think about, which is my measurement method doing a good job? And another way to ask that question is, how well do I know the limitations of the technology I'm using? And the answer in this sphere, and well, in general, is to take the MISEV guidelines to heart and use orthogonal techniques. So let's look at some examples. Because it's so popular still, let's focus on NTA for a second. This is still used by the majority of EV researchers, although people are, as I mentioned, understanding its limitations more and more. So let's do a little experiment with NTA, and you may have seen me present this before, but it's an important experiment, and if you haven't seen it before, feel free to try it yourself. It's very easy. So what we're going to do here is measure 3 different monodisperse samples, 150 and 94 nanometers diameter. And you can see that when NTA measures these samples each on their own, it does a fine job, right? It measures the peak amplitudes correctly, or the concentration correctly, and the diameters correctly. So that's great. And we would maybe conclude that the sensitivity limit of NTA is much less than 94 nanometers, right? It's doing fine here. And in fact, you'd be right for monodisperse samples. NTA can count as small as 10 nanometer particles accurately, as long as they're brightly scattering and monodisperse. The tricky part here comes up when we ask what happens in a polydisperse sample, like real world samples, right? So we can simulate that with these particles by mixing them together. And when we do that, this is the measurement that we see from NTA. So you can see here, the 94 nanometer particles are completely absent in the measurement of the mixed sample. And what we can conclude from this experiment is that the sensitivity of the method depends on the sample itself. That's a major problem when you don't understand in advance what's in the sample. Why does this happen? Well, the answer is basically physics. And it comes from the scattering intensity scaling with the diameter of the particles. So we're shining light onto these particles and we're detecting the scattered light that comes off of them. And the intensity of that scattered light scales with the sixth power of the diameter of the particle. That's a really strong scattering, really a strong dependence. So for example, a 400 nanometer particle scatters more than 50,000 times brighter than a 65 nanometer particle, even though it's not that much bigger. The tricky part is that we're in NTA method, we're trying to take videos of all these particles at once. And that represents an enormous dynamic range problem for the camera. And ultimately what happens is the large particles obscure the small ones in the image, and they can't be detected. So the sensitivity of NTA to smallest particles is obscured, is diminished in the presence of larger particles. When we repeat this experiment with MRPS, for example, though, because we're measuring particles independently of one another, same thing for the monodisperse samples. We measure each of the components accurately, but when we mix them together, we get what we expect, which is the superposition of each of the components that were added to the mixture. So this is an example where an orthogonal technique like MRPS adds a lot of value to the analysis of this sample. Of course, this sample is just a polystyrene bead example. So let's see what the implications of this are in real life. So for example, with a urinary EV sample. So here we'll take the perspective of a researcher that's analyzed this sample by NTA first. And we see a nice peak right around 130 nanometers diameter. This is a very promising data set. It's a nice clear peak, and we can track this and measure the concentration and stuff. That's great. You're following the MISEV rules though, and so you take this sample and measure it on a different technique also. So first, I will replot the exact same data on a log Y scale. And now we'll measure it with MRPS because we're lucky to have one in the lab. And when we do that, what we see is that the size distribution actually continues to increase all the way down to the smallest limit of detection of this technique. So in contrast to the peak that was shown by NTA, MRPS is suggesting that the concentration keeps increasing all the way down to smallest sizes, at least to 50 nanometers. So what's going on here? Well, luckily down the hall, we have a TEM that we can take the sample to. And when we do the analysis of the size distribution by TEM, which is the gold standard, we see excellent agreement between the MRPS and the TEM measurements. So what's happened now with hindsight, we can see that the sensitivity of the NTA method to these small particles in this polydispersed sample rolls off at smaller diameters. And ultimately, in the critical EV size range, it can be as large as 10,000-fold off in concentration. Remember, it's a log scale here on the y-axis. So this just emphasizes that orthogonal measurements give important information about our other methods and about what's really in the sample. So let's shift gears and talk about emerging technologies then. So one of the rapidly adopted ones is fluorescence NTA. This is just nanoparticle tracking analysis with a fluorescence measurement of the particles. So same optical method, and the size estimation still comes from diffusion, except we're using a fluorescence illumination instead. I'll also briefly discuss one of the coolest technologies I think that's out there, which is single particle interferometric reflectance imaging sensor, SP-IRIS, with and without fluorescence. This is, again, another optical method, although very different. It's not using diffusion. And it allows the identification and relative quantification of subpopulations in a sample, which is cool. Finally, flow cytometry is also recently being applied to EVs and small particles more. And this is another optical method. We'll discuss this at the end also. So first, fluorescence NTA. So in this case, now knowing the limit of detection is still critical, right? So let's look at this hypothetical example, similar peak profile, right? And you might be thinking now, well, we don't need the scattering intensity to be so strong now. That's not as important because we're looking at the fluorescence light. So even small particles that fluoresce brightly should be detected here. So this is probably a real peak, right? And so what I would ask you is this question. So is it that there are no fluorescent particles in this size range, the smaller one? Or is it just that they are too dim to be detected by fluorescence, right? So same question, different measurement technique. And the idea is, what is the limit of detection of the fluorescence signal? And so for accuracy, we need to know the limit of detection of size, like before, as well as the limit of detection of fluorescence. And that asks all kinds of questions about bleaching rate and things like that. So we need something better than this. Let's talk about SP-IRIS. So this is a cool method in which capture agents are immobilized on a surface. And so we functionalize the surface. And then we immobilize the particles from the sample onto the surface, specifically those that are captured by the antibodies. And then the technique uses interference imaging to measure the size and the relative counts on the different spots. So these are different spots here, right, illustrated. And if we add fluorescence to this technique, then we can co-locate the surface markers on the surface and get information about the different phenotypes. I really like this this technique because of the imaging power. They can image well below the diffraction limit of particle size, which is cool. There are some limitations to the concentration measurements from here, though. So in this process of the particles diffusing down to the surface, the diffusion time depends on size ultimately. So the time required for a particle to diffuse down to the surface is proportional to the distance squared. So that means that farther ones take quite a bit longer than close ones, but also to the diameter of the particle. And so let's put some actual numbers on here. So for 1 millimeter, about the thickness of a droplet, if you put it on the slide, a 50 nanometer particle takes 4 hours to diffuse that distance. But a 200 nanometer particle takes, a good part of a day, 16 hours. And so that's why the incubation times for binding your sample onto these surfaces are long. The issue comes when we talk about relative size concentration. So the large particles in this case are ultimately underrepresented on the surface because they take way longer to get to the surface and interact with the surface than the smaller particles. And so for any kind of surface immobilization process, we're limited to relative concentrations of the different sizes and different phenotypes, right? This doesn't, yeah. So let's move on also and talk about flow cytometry. So this is where I think the future lies. This is a super exciting area for EV analysis. There are key advantages to using flow cytometry. It's a foundational technology for large particles and cells, right? It's ubiquitous, it's well characterized. The particles are analyzed in situ, so without any mobilization required. And the method is compatible with standard protocols and labeling processes, SOPs. And that's largely because many of them were developed specifically for flow cytometry. So in this method, we illuminate particles one by one with laser light, and we collect the light in a few different ways. First, we collect the scattered light, so the forward scatter and the side scatter, for example, and those tell us information about the particles. And also, we can collect the fluorescence emission light. And nowadays, flow cytometers are almost spectrometers, right? They have so many detection channels available. And we trigger the measurement based on one of two options. We can use the scatter pulse to detect the particle, or we can use a fluorescence pulse to detect the particle. So with scatter detection, again, we have the same limitation before, where the intensity of the scattered light it really falls off quickly with diameter. So the smallest particles are very weakly scattering. Plus, EVs are low contrast with the surrounding medium, so that makes an additional challenge. Ultimately, that means that the conventional flow cytometers have limits of detection by this method of 100 or 200 nanometers in diameter. The alternative is to use the fluorescence pulse to detect the particle. And in this case, you can detect smaller particles as long as they're bright, right? But again, there's a dependence on the detection or the signal amplitude, because the number of labels that you can bind onto the surface of these particles decreases with surface area. So if you're relying on a large fluorescent signal in order to detect the particle, then that gets harder and harder to generate with smaller particles because you can't put so many fluors on there. And then there's a question, what about size? So to get the size information, we need to use the scattered light. And in this case, we need to also assume the particle index of refraction. So that's a key assumption that starts to break down for heterogeneous samples where there are particles of different index contrasts in the sample, and so you're not quite clear what you're measuring. It's possible to correlate the fluorescence amplitude with size, and I know there are new techniques being developed for doing that in a rigorous way, particularly by John Nolan and Celarcus Biosciences. So these are cool emerging technologies, and they use specialized assays to correlate the fluorescence brightness with like a lipid marker, a membrane dye with EV size. So this is where I feel like the future is, and still some work to go to get to the smaller sizes. So what is next, and what's needed, right? For accurate size and concentration and single particle phenotyping. For just concentration and size, we have various techniques available, for example, Spectradyne's MRPS. In order to get to more in-depth EV analysis, we need size and concentration, but not just counts, with phenotyping. And we still have a critical need in this area. So what I believe we should be aiming for as a field, technology field, is single particle phenotyping with accurate size and concentration, with well-characterized limits of detection. So size, concentration, and fluorescence intensity, for example, in MISEV units. And of course, it should be fast and easily integrated with standard methods and reagents. All right, so I hope that I have encouraged you to think about a few different things during this talk. First of all, that you should take size and concentration measurements seriously. And that's essentially because concentration is the dose or a critical experiment of experimental parameter that needs to be controlled. 2, is my measurement method doing a good job? Hopefully, you've been encouraged to think about what the limitations of the technology are and the value that an orthogonal technique brings to bear on experimental questions. And finally, hopefully, you've been able to ask, what do we need next? And in our opinion at Spectradyne, it's accurate quantification of particle subpopulations in complex samples, right? that's where the field is going. So stay tuned. If you would like to learn more about Spectradyne and its technologies, please visit our website. In our library, we have publications, from independent third parties, so you don't have to take my word for any of this. We encourage you to come and read it in our library. We've got application notes and videos and product specifications all on our website. If you're interested in learning more about our technology, we encourage you to please send us a free sample, and we will measure it for you for free. And come and speak to us today at our booth here at the conference at Coronado Island. Thank you very much for your time, and I hope to hear from you soon.